Okay, welcome everybody to today's webinar. A few housekeeping things before we get started. We'll try to get to any questions that you may have at the end of the presentation. If you do have a question, please type it into the question area within the GoToWebinar navigation, and we'll get to as many as we can in our allotted time. Within the next 48 hours, one continuing education credit will be emailed to all participants on the live version of the webinar, and a link to a recorded version will also be available. With that, I'd like to turn it over to our ACSM moderator, Dr. John Jakisik, an ACSM Fellow and Professor and Chair of the Department of Health and Physical Activity at the University of Pittsburgh.
Great. Good afternoon, everyone. My name is John Jakisik, and on behalf of the American College of Sports Medicine, I want to welcome you to this webinar entitled Blending Proteins to Build Muscle, What Does the Research Tell Us? Our co-sponsors today, the American College of Sports Medicine and DuPont Nutrition and Health, have a strong relationship that is built on providing the best and most useful information to individuals participating in nutrition, exercise, and training programs. The ACSM is the largest sports medicine and exercise science organization in the world, with more than 50,000 members and certified professionals worldwide. The ACSM is dedicated to advancing and integrating scientific research to provide educational and practical applications of exercise science and sports medicine. DuPont Health and Nutrition and Health combines in-depth knowledge of food and nutrition with current research and expert science to deliver unmatched value to the food, beverage, and dietary supplement industries. Today's webinar, presented by Dr. Blake Rasmussen, is presented by Dr. Blake Rasmussen. Dr. Rasmussen is the current chair of the Department of Nutrition and Metabolism at the University of Texas Medical Branch in the School of Health Professions. Dr. Rasmussen holds the Lloyd and Sue Ann Hill Endowed Professorship in Healthy Aging and is currently the director of the department's Muscle and Biology and Metabolism Laboratory and the leader of the NIH-funded Claude D. Pepper Older Americans Independent Center Pilot and Exploratory Studies Corps at UTMB. After earning his BS and MS degrees in exercise science from Utah State University and a PhD in zoology/cell biology from Brigham Young University in 1997, he came to UTMB in 1997 as a postdoctoral research fellow in the Department of Surgery and Metabolism. Completing his training in 1999, he then accepted an assistant professor position at the University of Southern California in 2000 before returning to UTMB in 2004 as an associate professor in the Department of Physical Therapy in the School of Health Professions. He received tenure in 2006. Dr. Rasmussen's research is focused on how nutrition and exercise influence muscle biology and in developing clinical interventions to prevent muscle loss during aging and other muscle wasting conditions. He's published over 100 papers in the field of nutrition, metabolism, and exercise physiology, and has trained several postdoctoral students and PhD students. It's with great pleasure that I introduce to you Dr. Blake Rasmussen.
Thank you, Dr. Jakisik, for the kind introduction. Today I'd like to discuss some of the work we've done over the last few years with DuPont Nutrition and Health, looking at blending proteins as a protein supplement for muscle biology and for muscle growth. And so my outline will be the mechanisms underlying how exercise and protein ingestion increase muscle protein synthesis. I'll then talk about differences in protein sources and look at the effect of a protein blend on the mTORC1 signaling pathway and muscle protein synthesis in acute and prolonged exercise studies in humans. And then I'll finish with some conclusions. So over the past several years, we and other groups around the world have been looking at protein synthesis in response to exercise and in response to nutrition. And when I say nutrition, for the purposes of this presentation, I'll be referring to either protein ingestion or essential amino acid ingestion. So normally, as you can see here on this figure, when you exercise an individual, basically performing leg extension, muscle contractions, a normal young healthy adult will have about a 50% increase in muscle protein synthesis rate. If you give that individual essential amino acids, shortly after they do the exercise bout, you can essentially double the protein synthesis rate. So you get an additive effect of the amino acids and the muscle contraction. I'll talk more about aging a little bit later, but we have a problem with aging in that older adults tend to have a much reduced response to exercise for stimulating protein synthesis, but that can be restored by providing protein supplements after the exercise bout. So before I get into some of the research we've done, I think it's important that we discuss some of the mechanisms behind how exercise and protein ingestion might increase protein synthesis and what we know about that. So this is kind of a complicated slide, but if you look at the top here, this is the blood. When you eat or after or performing a bout of exercise, amino acids enter the bloodstream, particularly after eating or after feeding, and cause a release of insulin. So insulin binds to its receptor on the endothelial cell, which lines the capillaries near the muscle, for example, and causes vasodilation through a nitric oxide induced pathway. The other thing is that amino acids in the bloodstream have to cross this endothelial cell barrier as well, and so they enter the endothelial cell through their specific amino acid transporters, and then they're exported from the endothelial cell on the other side through transporters as well. They then cross the interstitial space, where they are then transported through transporters again into the muscle cell, where they can activate this pathway that we're going to refer to as the mammalian or mechanistic rapamycin pathway. Activation of this pathway leads to phosphorylation of downstream events that eventually lead to translation initiation and elongation and an increase in protein synthesis. So we're learning a lot, especially in the last 10 years, about how amino acids are able to come in from the blood, cross the endothelial cell, enter the muscle cell, and activate this pathway to turn on protein synthesis. Another pathway is the insulin pathway. Also, insulin needs to cross the endothelial cell barrier and bind to its receptor on the muscle, and then activates a very similar pathway, but we refer to this as the AKT, or insulin receptor pathway, and it also activates mTOR, but from a different point of view as amino acids. Now, muscle contraction also activates mTORC1, but it does through a different pathway as well. So we have three independent pathways all converging on mTORC1, and I'm going to talk more about mTORC1 later in the presentation, but I wanted you to keep an eye on this protein and knowing that it is considered one of the key cell growth regulators in most muscle cells, and is important when we're trying to design evidence-based products to activate protein synthesis and eventually muscle growth. So some recent work just within the last few months has identified the actual pathway of how amino acids turn on protein synthesis. And so we've known for many years that the branched chain amino acids, and in particular leucine, seems to be the most potent of the essential amino acids for activating protein synthesis. And it does this by activating mTOR through a unique pathway where it binds to a protein called cestrin-2, which inhibits its activity of Gator-2 and leads to this complex where mTORC1 is then translocated from inside the cell to the lysosomal membrane, where it is then becomes active. I'm not going to go into all the details of this, but I wanted to highlight that this pathway is new. We're just learning about it now, but have now identified what we think to be is the main pathway for how leucine turns on protein synthesis. Now, just within the last month, another protein complex has been identified called Castor-1, which has a very similar effect, but it responds to arginine, so it's an arginine sensor. So we have both leucine and arginine sensors within the cell that turn on mTOR, and this is how our muscle cells respond to amino acids and how they turn on protein synthesis. We know mTOR is important. We know that it's required because if we give a drug that's called rapamycin, which is a specific inhibitor of mTOR, before we have somebody exercise or before we feed them a protein we can completely block protein synthesis and block the pathway. So we know this mTOR pathway is important. And these two slides are just a background information of some of the mechanisms behind why we're designing these protein supplements to turn on protein synthesis and eventual muscle growth. So the first question, and a question I get often, is what protein is best for promoting muscle growth? Well, to answer that question, it really depends on the quantity of the essential amino acids that are contained within that particular protein, and especially leucine. And so the gold standard in the field for many years has been whey protein, because whey protein has a high proportion of essential amino acids, and it has a very high proportion of branched chain amino acids in particular, and leucine. However, other protein sources like casein protein or soy protein isolate also are considered high-quality proteins, and they do have high amounts of branched chain amino acids and leucine, but just in different proportions. So one of the things we wanted to look at was blending proteins, and why would we want to blend proteins for a protein supplement or for sports nutrition? And so a couple of things. One, there's a lot of people that are interested in replacing some of their animal protein with vegetable protein, for instance. Other people want to take advantage of some of the potential health benefits that are associated with plant proteins. Another interesting finding that some work that DuPont has done has identified that the taste, when you combine blended proteins with dairy proteins, or basically soy and dairy listed here in blue, is that the people that are involved in these research studies support, they score the blended proteins better for taste. And so that's a good example of why these proteins can be blended and taste better for the consumer, and also contain the right amount of amino acids. Now on the right side of this slide, I wanted to highlight a difference between a couple of the standard high-quality proteins that are used in sports nutrition. And the gold standard in the field, as I mentioned, is whey protein. And if you take one scoop of a typical whey protein product, it contains about 20 grams of protein and about 1.9 grams of leucine. Now, if you take the same 20 grams of protein from a soy protein isolate, you'll see that it's slightly lower amounts of leucine, 1.5 grams in this example. And so we have done a lot of work, and so have other groups, suggesting that there is really a leucine threshold for maximizing muscle protein synthesis in young healthy men and women. And that seems to be around 2 grams of leucine in a protein supplement. So about approximately between 1.8 to 2.2 grams of leucine seems to be the trigger point, or the threshold that's necessary to actually maximize muscle protein synthesis. And so taking advantage of that knowledge, if someone wanted to use a soy protein isolate, for example, and rather than whey protein, what they would need to do is to increase the number of grams by 7 to equal the 1.9 gram of leucine that you get in the wheys, which would be an additional 28 calories. So that's just kind of an idea of when we design some of these protein supplements, we're taking advantage of our knowledge of leucine being a key activator of the mTOR pathway in protein synthesis. We know exact kind of pretty precisely how much leucine is required, close to 2 grams in young adults, and so that's important when you decide to develop an evidence-based nutritional product. A couple other things we looked at when we were designing some of these protein supplements was that we noticed that if you look at some of the standard complete high-quality proteins in the field, whey, soy, casein, that all are considered a high-quality protein, all have essential amino acids, and all have a PDCAAS score of 1 or more. So, but there's a few things that are different. For example, whey protein has a high digestion rate, soy protein has an intermediate digestion rate, and casein has the slowest digestion rate. All have been shown in various studies to be supportive of gaining lean mass during exercise training. Whey has the highest proportion of leucine, soy has the highest proportion of arginine and glutamine, and also contains some antioxidant activity. So basically our theory or our hypothesis behind this, is that if we combined all of these proteins into one supplement, we could actually put a check mark in each one of these categories. So to begin doing this, we did a preclinical study in rats, where we did this in combination with DuPont Nutrition and Health, where we wanted to develop a variety of different blends and see which blend might be the most effective at promoting protein synthesis. And so to do that, we initially started with animals, and then we worked our way into doing 2 studies in a young group and then an older group, and then a prolonged study. And I'll talk about all of these as we go on today. So our preclinical study was the basis behind developing this blend. And what we wanted to do here is to evaluate the ability of a whey protein isolate and soy protein isolate alone, as well as in different combinations of 2 blends to see how they affected muscle protein synthesis. So male rats were trained to eat 3 meals per day. The test meals were similar among groups, although they varied in the leucine content. And then we measured fractional synthetic rate with stable isotope tracers. And here's the groups that we looked at: whey protein isolate, casein, soy protein, and the different blend combinations. And this was published in Clinical Nutrition a few years ago. And so what we found, if you look at this figure on the right, was that we looked at the blend 1, the blend 2, the soy isolate, the whey isolate, and we looked at the area under the curve for muscle protein synthesis. And we identified that blend 2 produced the greatest area under the curve for protein synthesis. And if you go back 1 slide, you can see that blend 2 consisted of 25% whey, 50% casein, and 25% soy protein. So we then decided to translate our preclinical study into a clinical study, where we looked at this protein blend, blend 2 in this case, following resistance exercise humans. And this is Paul Reedy, the picture here on the right, and he did this project for his PhD dissertation project. So the aims of this study were to determine whether a soy-dairy blend promoted muscle protein synthesis to the same or better level than the gold standard in the field, which is the whey protein isolate. We also then wanted to know, would the amino acid net balance across the leg, in this case the muscles of the leg, used as a marker for overall protein muscle anabolism, would be more positive when consuming the soy-dairy blend versus the whey alone. And you can find more. These 2 aims were addressed in these 2 papers listed here in the lower right. So subjects performed a high-intensity resistance exercise, which consisted of 8 sets of 10 repetitions. And then they had catheters placed. We used stable isotope tracers here on the bottom to look at muscle protein turnover. We had a resting period where we took muscle biopsies and we took blood samples. Then we had them do exercise. The exercise took about 40 to 45 minutes to complete. That's the 8 sets of 10 repetitions here. 1 hour after the exercise bout, they either consumed the protein blend or they consumed the whey protein isolate. And here's the composition of the 2 intervention supplements here. The protein blend contained 19 grams of protein. The whey supplement contained almost 18 grams of protein. And they were matched pretty much on the leucine threshold that we talked about earlier, were 1.8 grams of leucine in the blend and 1.9 in the whey. And here's those supplements again. And the blend, as I mentioned before, contained 25% of whey, 25% of sucrose soy protein isolate, and 50% of caseinate. And the whey was 100% whey protein isolate. Our subjects were young, healthy men and women. When we looked at the net balance across the leg, this is the protein synthesis rate minus the protein breakdown rate to give you an overall net balance across the leg. We see that net balance was very positive in the gray whey group, and it went up very quickly following ingestion of the drink after exercise, where the blend group showed a more prolonged and elevated time course as compared to the whey protein group. So you can clearly see that the addition of two slower digestion rate proteins in the blend produced a more prolonged net balance across the lake. When we looked at mTORC1 signaling, this is the pathway that I discussed at the very beginning. These are the phosphorylation of two of the downstream effectors of the mTOR pathway, S6K1 and ribosomal protein S6. You see that the increase in phosphorylation rate went up in both groups to a similar extent, indicating that the mTOR pathway was activated in both the blend and in the whey group. If you look at this figure, this is the branched chain amino acid, total branched chain amino acid concentration on the axis here, and then this is protein synthesis on the right axis. And you can see the whey is in gray. It has that very rapid increase in branched chain amino acid concentrations and then comes down rather quickly within 2 hours. Whereas the concentration of the branched chain amino acids in the blend group goes up and stays elevated for a longer period of time. When you look at protein synthesis, these are the histograms. You'll see that muscle protein synthesis goes up in the early period post-exercise in both groups. And in the longer 3 to 5 hour post-exercise period, you'll see that protein synthesis is still elevated in the blend group and is not quite significant in the whey group. However, there was no significant difference between the groups at this time point. So the soy-dairy blend versus the whey protein alone provides a prolonged delivery of amino acids to muscle, making it optimal for consumption following resistance exercise. So we then wanted to extend it into a clinical study into young adults, doing a long-term study, and I'll talk more about that at the moment. But I wanted to then determine whether this same effect of the protein blend, we wanted to know whether we could find it as well in older adults. And so we'll come back to the chronic study in just a moment. But first of all, we did a study in older adults where we gave this exact same blend to older men, and this was a dissertation project by Michael Borak in the lab. So the aims of this study were very similar. We wanted to know whether or not this protein blend would prolong blood hyperaminoacidemia in older adults, whether it would extend skeletal muscle protein synthesis as we saw in the younger adults, and whether it would improve muscle protein turnover. So we hypothesized that the different digestion rates of the proteins comprising the soy-dairy blend would result in a more sustained hyperaminoacidemia as compared to whey, and that this prolonged hyperaminoacidemia would lengthen the muscle protein synthetic response for the protein blend as compared to whey. So we collected data on 19 older men between the ages of 55 and 75. They were generally healthy, had a BMI less than 30, and they participated in the same 8 sets of 10 repetitions of leg extension exercise as was described for the young adults. We had a very similar protocol for our study design, where we used the same amino acid tracer. However, we added an additional tracer to look at muscle protein breakdown in addition to protein synthesis in this case. We took muscle biopsies as indicated by the thick arrows here at rest and in the early and late periods following exercise. They performed the bout of exercise after the resting period. And then the only real difference was that we changed the dosage of our protein, because a lot of work from groups like from Stu Phillips' lab and from Luc Van Loon's lab have identified that perhaps we need a bit more protein in older adults to have the same effect as we see in younger adults. And in fact, a lot of work is now showing that perhaps the leucine threshold is elevated with aging, such that young adults can maximize protein synthesis at 2 grams of leucine within a protein supplement, but perhaps that is elevated to as high as 3 grams of leucine in older adults. And so what we did for this particular study to ensure that we could see an increase in protein synthesis was that we gave 30 grams of whey protein and 30 grams of the protein blend to older men following exercise. Now, this time we didn't match for leucine. We let the dosage fall out as it may, And in this case, we had about 3.26 grams of leucine in the whey group and about 2.8 grams of leucine in the protein blend group. So when we look at the amino acid concentrations in following the exercise bout and following the ingestion of the protein, we see a little bit different response. So the normal prolonged effect that we saw with younger adults was not seen with older adults, and this is kind of a common event that we see with aging, is that the blood amino acid concentrations tend to be already a little bit elevated in older adults. So we saw an increase in blood hyperaminoacidemia, but no difference between groups as we saw with young. If we look at the leucine area under the curve during that full 4 or 5 hours post-ingestion, we see that the whey protein supplement was slightly higher, but this was not significant. But we had a significant increase in leucine area under the curve in both groups. If we look at mTORC1 signaling, I'm showing here the same two proteins I showed before, S6K1 phosphorylation and S6 phosphorylation. We see a similar increase in both groups. So what this slide represents is that mTOR activation was turned on in both the protein blend group as well as in the whey protein group in older men. When we look at protein synthesis, we see a significant increase in the whey group. There was an increase in the protein blend group. It did not quite reach significance because of the elevated protein synthesis rate at rest that was detected in our protein blend group. However, there was no difference in this elevated protein synthesis between groups following the exercise bout, indicating that protein synthesis was elevated in both groups to a similar level. Muscle protein breakdown. This is something new we looked at for this particular study. And we saw that there was a reduction in muscle protein breakdown in the post-exercise period following ingestion of the blend or the whey protein group. And the reduction was similar between groups. So in summary of these two acute studies in men and older men and younger adults, we found that the protein blend ingestion prolonged hyperaminoacidemia in young adults as compared to whey protein isolate, whereas hyperaminoacidemia was similar in older men following ingestion of both groups. The protein blend and the whey protein ingestion activated mTORC1 signaling to a similar extent in both young and older men, and the protein blend and whey protein ingestion resulted in a similar and elevated post-exercise muscle protein synthesis rate. So this led us to conclude that use of a soy-dairy protein blend may be a useful nutritional strategy for maintaining muscle mass and function during aging while reducing the disability associated with sarcopenia. So it's a potential other source of a high-quality protein supplement that could be used to combat sarcopenia with aging. So then we wanted to know, we saw some positive results for both our young men and in our older men, indicating that following exercise, a protein blend or whey protein was capable of activating the mTOR pathway and turning on muscle protein synthesis. But these were one-day studies, so acute studies, and so we wanted to know whether or not, if we did this over several months of exercise training, whether we could enhance muscle growth. So to do that, we wanted to look at the literature first to see what kind of increase in muscle mass should we be expecting to see with protein supplementation and exercise. And so there was a nice meta-analysis that was performed by Luc van Loon a few years ago, and he had very strict criteria for what studies he wanted to include in both groups. And based on the criteria he had for this particular paper, they identified 16 studies that they included in the meta-analysis in younger adults and 6 for older adults. And if you look at, this is the kilogram of lean mass gained beyond exercise alone, you can see that in our younger group that it was an addition of about 0.69 kilograms of additional muscle mass gain based for the protein supplementation, where in our older group it was about 0.47 or so. So and the combination was about half a kilogram expectation of an additional increase in lean mass above and beyond exercise alone. So I think a couple things to take from this is, one, the protein supplementation is capable of enhancing lean muscle growth, but it's not going to have a super large increase on lean mass. It's favorable, promotes a little bit more lean mass, but the effects are rather modest. So we decided to look at a chronic study where we gave the protein blend and the whey protein isolate during 12 weeks of resistance exercise training. And this paper is also currently under review and is not published as of yet. So what we decided to do was to recruit 60 young men into 3 groups. And we wanted to put 20 young men into a protein blend group, 20 into a whey protein isolate group, and 20 into a placebo group, which was a caloric maltodextrin eucaloric placebo group. To do that, we contacted 422 people, screened several, and then through our randomization process, we tried to put about 22 or 23 people into each group in order to get our 20 at the end, based on a few dropouts and a few other things that took place during the 12 weeks of training. We finished with 22 people in our protein blend group and 18 in both the whey protein group and the placebo group. So our aim of this study, which was a double-blind, parallel design, randomized clinical trial, was to determine the effect of a soy-dairy blend during a supplementation with this soy-dairy blend during the resistance exercise training on lean body mass, muscle thickness, and strength in young active men. So a couple of our primary outcomes, we looked at, to look at muscle mass, we looked at lean mass through DEXA, and we also used ultrasound and microscopy to look at muscle cross-sectional area. For muscle strength, we looked at 1 repetition max, and then we used a Biodex isokinetic dynamometer to look at muscle strength, torque, power, so forth. Whoops, I'm going way too fast here. Sorry. So here is the run-in before the study, the 12 weeks of training, and how we set up the repetitions and sets, and how we used 8 different groups, muscle groups, and how we progressively increased the resistance of training throughout the exercise bout. So by the time they were into their last month of training, they were performing their bouts to failure and at about approximately 80% of 1 repetition maximum. And then what we did was we gave the supplements immediately following the workouts, which were 3 days a week. And then we also gave the protein or placebo supplements between meals on the non-workout days, or 4 days a week. So our supplements were here, as mentioned before. We had the placebo group, which is maltodextrin, the protein blend, which is the same blend we've been discussing, which is 25% whey, 25% soy, and 50% caseinate. And then 100% whey protein isolate was the comparator group. So if we look at the content, macronutrient content here, we can see that the protein groups were about 22 grams of protein in both groups. And the calories were just under 100 calories for all 3 groups, and they were pretty much eucaloric. If you look at some of the essential amino acids in the product here, you can see that leucine was over 2 grams, was 2 grams in the protein blend group and 2.3 grams in the whey protein group, with none, of course, in the placebo. So looking at muscle strength, we saw all 3 groups had a significant increase in isometric knee extension strength, knee flexion strength, isokinetic knee extension strength. There was a large increase in all groups. So essentially all 3 groups showed similar increases in muscle strength. When we look at lean mass, here's pre to post changes in kilograms for lean mass. You'll see protein blend in the black bar, the whey protein in the gray bar, and the placebo group in the light gray bar. Total lean mass for whole body showed nearly a 3 kilogram increase in lean mass for the protein blend group, and a 2.5 kilogram increase for the whey protein group, and a 2 kilogram increase for the placebo group. There was a trend, but not significant difference between this increase in lean mass between the blend and the placebo. and between the whey protein and the placebo. It seems like some of the lean mass was higher with the blend in the arms, for example, as here, as well as the legs and the trunk tended to have similar changes in lean mass over the 12 weeks of training. If you look at some of the individual changes in lean mass, there does seem to be some interesting things that pop out with the soy-dairy blend. Subjects receiving the soy-dairy protein blend exhibited a trend for increased lean mass compared to the control. I just pointed that out on the previous slide. However, there was no difference in lean mass between the whey protein isolate and the control, and the inter-individual variability was quite evident. And that's shown on this particular figure here. Here's the change in lean mass on the y-axis. The placebo group, here's that 2 kilogram increase in lean mass, the 2.5 are in the whey protein here, and the 3 kilogram increase in the protein blend group. One thing, these are all data points, so you can see there's a wide range of individual response to the exercise training and supplementation. One thing, if we look at the literature, the expected gain in lean mass is about 1.5 kilograms of lean mass from exercise alone. And so anything above and beyond this would be due to the protein supplementation. You can see we had about a 2 kilogram increase in our placebo group, which is similar to that 1.5 kilogram, but there was a wide range of responders and non-responders. However, if you look at those people that were above this 1.5 kilogram line, the expected gain, the protein blends were significantly higher, the number of people that were above this threshold. It's indicating that the protein blend was producing more responders and producing more people with a higher increase in lean mass. What's also interesting is that if you look at the lean mass changes from the time 0 to 6 weeks into training, the midpoint versus midpoint to post, there start to be a leveling off in the whey protein group and the placebo group for changes in lean mass, whereas there continues to be an increase in lean mass in the protein blend group. So only the soy-dairy blend had a significant increase in lean body mass during the last 6 weeks of resistance exercise training. If we look at muscle thickness, this is muscle thickness of the vastus lateralis on the thigh, we see a very similar increase in muscle thickness for all 3 groups. There was no difference between the protein blend or the whey protein or the placebo group. But there was an increase in all groups. There was also a similar increase in strength for 1 RM for squats, knee extension, and bench press, and all the gains were very similar to what has previously been published in the literature. If we look at muscle cross-sectional area, taking a muscle biopsy sample, looking at the cross-section of the muscle sample from the vastus lateralis, we show the same thing as we showed with the ultrasound, and that is there is an increase from pre to post training in the placebo group, the whey protein group, and the protein blend group. And these were all very similar changes with no differences between groups. So then we wanted to look at a few other things. One of the things we noticed was that there's quite an extensive literature on the cholesterol lowering effects of soy protein in humans. And so we wanted to look at how this might affect cholesterol levels during training in our young men. And what we found was that the soy dairy blend significantly reduced total cholesterol, LDL, and VLDL, and triglyceride levels from baseline following 12 weeks of training and blend supplementation, whereas the whey protein group only had a reduced total cholesterol from baseline. And the total cholesterol to HDL ratio was significantly lower following the blend supplementation as compared to the whey. So soy dairy protein blend improved blood lipid profile, independent of the improvement associated with exercise alone. One confounding variable in our study in particular was that our subjects were on the standard American diet, which is pretty much a high protein diet. So all of our subjects in both the protein groups as well as the placebo group were ingesting 1.2 to 1.3 grams per kg per day of protein. So they were already taking a large amount of protein. And so any addition of the supplement from the blend group or the whey protein group, the effects from the supplement were somewhat minimized because of the already high protein diet from our individuals. So we then wanted to look at all the papers that were ever been published on protein and exercise training in both acute exercise studies and chronic exercise studies. And this resulted in a large critical review paper in the Journal of Nutrition just this recent February. And so I will point you to that paper to take a look at, but in that paper you'll find it's 28 pages in length and it covers every paper that's ever been published. And it also has a large amount of supplementary materials with tables and figures and Excel spreadsheets that would be handy for anybody interested in looking at all the work that has gone into this particular topic. One thing we wanted to highlight here, and this is just kind of a summary from the paper, is that if you take someone and do weight training exercise for several weeks to several months, there is an effect with protein supplementation on enhancing lean mass. It's quite minimal compared to this is a carbohydrate placebo control group, but there is a difference of about 0.63 kilograms of extra lean mass gain by adding protein to your training protocol. And so this is very similar to the meta-analysis that Dr. Van Loon's group had previously published in a smaller amount of papers. But you can also see the difference. These are individual responses from some of these studies. And you can see there are a larger amount of high responders in the protein group as compared to the placebo group. So I point you to that paper for a lot of good resources on this particular topic. I'd like to summarize by saying that exercise and protein ingestion increase the muscle protein synthesis by activating the mTOR pathway. Protein supplementation during exercise training can enhance muscle growth. However, this effect is small and dependent on total protein intake. High-quality protein sources from both vegetable and animal sources produce similar increases in muscle protein synthesis and growth during resistance exercise training, or plant sources and animal sources. Specifically, some conclusions and summary on the blend that we've been looking at in comparison to whey is that we find that the soy-dairy blend supplementation demonstrated a potential for maximizing the acute adaptive growth response of skeletal muscle to resistance exercise. It also extended or in our long-term study, the soy-dairy protein supplementation enhanced lean mass gains after resistance exercise, yielding a greater percentage of subjects that experienced gains above the expected threshold, while also improving blood lipid and lipoprotein concentrations. And high-quality soy protein provides other benefits that go beyond muscle and makes important contributions to overall health and wellness across the lifespan. So with that, I'd like to acknowledge the funding sources for our studies and the students and fellows that have participated in all of these studies. So thank you.
Thanks a lot, Blake. John, are you still there to take some questions for us?
Yes, do you hear me, Nate?
Yeah, we're good. Go ahead and offer up some questions for Blake if you could.
Yep, I do have a few questions. Blake, if you could maybe think about this from a little bit more of a clinical application perspective for us. I mean, this is great work, real exciting. And a question that arises is, you know, when you start to think about this resistance exercise, it seems like in most of the trials that you've done and have cited, the resistance exercise is a relatively vigorous amount of activity. And I just wonder, is there a minimum threshold of resistance exercise that maybe we need to be thinking about, in which we might be able to see some of the benefits of these proteins that you're talking about? Or does it really have to be at this high level? Do we have any evidence around that issue?
That's a good question. I'm not aware of any evidence on that, to be honest. and I think most of these studies have been designed in order to see whether or not you could get any enhancement in strength gains or muscle gains. And to do that, you really need to propose a kind of a higher intensity resistance exercise and progressive resistance exercise training protocol. But that is a good question, and I think it might be have a lot of relevance, especially for our older adults and for looking at sarcopenia, as to whether or not there is a physical activity threshold, for example, that either would help prevent the loss of muscle mass or may work in combination with protein supplementation to enhance lean mass. But as far as I know, I'm not aware of any studies that have looked at that.
Yeah, I think that's I think it's interesting for the folks who are on the line here. In that, you know, if especially as you mentioned, the older adults, it may be difficult for them to maybe engage in that higher intensity activity that we're talking about, but maybe they could get away with what I'll say a little bit less exercise with the right protein supplementation to still have benefits. So I think there's some interesting work there that probably needs to be done. Another question that has arisen is, and early on in your presentation you talked about, and then throughout you talked about this idea that many of these proteins and amino acids need to be ingested within, you know, a certain period of time following the exercise bout to start to see kind of these benefits. Again, kind of thinking clinically about that, and you know, and just listening to our patients and participants talk about these things, they pay attention to that, but those who are less educated think that it's just okay to consume these things at any time during the day and don't really time it with their exercise. I'm wondering if you might be able to comment a little bit about how that there's either a good thing, a bad thing, or kind of an indifferent thing. Are they just kind of wasting their money on these things, or is there still some benefit even if they don't time it appropriately with the exercise?
Yeah, that's a good question. You know, when we started these studies about 15 years ago, many of us in the field kind of looked at the old data from the 70s and 80s on glycogen repletion with carbohydrate supplementation. And so we just started to design a lot of our studies as to whether or not, you know, giving amino acids or protein before exercise, during exercise, right after exercise, within the first hour or two after exercise, or later would be beneficial. And it took a few years, but almost all of the literature now suggests that it's best to consume these proteins or amino acids in the post-exercise period. And so within the last few years, though, we've also been looking at the effect of exercise. We know that exercise increases insulin sensitivity for 24 to 48 hours post-exercise, but we know very little about how that might affect protein metabolism. And so a lot of the more recent research is suggesting that in a similar vein to the change in insulin resistance or improvement in insulin sensitivity, that our muscles become more efficient, at least for 24 hours post-exercise. And so as long as so kind of the thinking is not so much that there's an open window of opportunity, as some people have suggested, for consuming these nutrients right within these first few hours after exercise. Rather, the thought now is changing, that perhaps we need to look at maybe distributing our protein evenly throughout the day following a bout of training, in order to take advantage of the fact that our muscles become more sensitive, they are more efficient at taking up the amino acids and using them for protein synthesis, muscle repair, muscle regrowth. And so that kind of goes along with what we know about how muscle grows, how it repairs itself, how hypertrophy exists, is that there needs to be amino acids present to build muscle protein. And so the thought now is not so much of this window of opportunity for protein in particular, but rather making sure that we have sustainable and enough protein at our 3 different meals following the training bout.
Yeah, very, very, very interesting. And so I have one last question, and before the webinar ends, and this may be kind of asking you to reach a little bit, but I know you've done a lot of work in this area of, you know, aging and sarcopenia and so on. And as we think about this, you know, we have kind of the healthy older adult and the kind of the less healthy older adult. In your take on this, is there a point in the aging process whereby which maybe we've gone over the threshold where possibly, you know, the addition of the protein or the addition of the protein in combination with the exercise has kind of, we've hit our threshold by where it's not going to have the effect that we've thought. Have you seen any of that where the person has just kind of deteriorated to the point where you almost can't recover them at that point in time? Any thoughts around that?
Well, most of our work has been done in fairly healthy older adults in their 70s. And in that case, providing a little bit more protein or providing the protein following exercise seems to have a very helpful effect on the acute muscle growth response. However, we've done a couple of studies, and we're currently doing an ongoing clinical study based in much older individuals in our acute care for elders clinic. And Rachel Deere is the postdoctoral fellow who is working on this right now. And what she is doing is taking these older adults that have been placed into this acute care facility for a few days, mostly because of pneumonia or some other illness, and they get placed into the hospital for 3 or 4 days. They lose a lot of muscle due to bed rest and hospitalization, and then they get released from the hospital. And so she's doing a study to look at what is the retention rate or what is the readmission rate of these older adults if we exercise them, or if we exercise, and this is not like, really strenuous exercises we do with younger people, but, or providing protein to them. And her preliminary data suggests that the nutrition is more helpful than the exercise at this point in helping prevent these older adults from being readmitted to the hospital. So, but this is very preliminary work, and so we're trying to work on that, and there's just not very much data available on real frail older adults, and whether or not this threshold maybe deteriorates in really old people.
Right. Yeah, very, very interesting information, and I want to thank you for your time and expertise today, Blake. I mean, I think this is very, very rewarding for everyone who has been a part of this, and I think it not only has been informative, but it really opens up the opportunity for where we can actually make some impact, and also some things that we don't know at this point in time, which I think is fantastic for all of us. So with that, I will ask Nate from the American College of Sports Medicine if there's anything else that we need to do, but otherwise we will be closing out this webinar today. Nate, is there anything else?
Nope. So just on behalf of ACSM and DuPont Nutrition and Health, thanks to everyone for attending. This will conclude the webinar.
All right. Thank you.
Thank you all.